This part of the thesis were already published in the respective journal Biochemistry. The remaining two parts of the thesis deal with UP, which could be an interesting starting point for the production of modified nucleosides to be applied in anti-cancer and antiviral therapies. Since thermostable proteins have some advantages for industrial use, Mr. Castelline has isolated genomic DNA from the hyperthermophilic archaean Pernix and amplified its UP gene by PCR. The gene was then cloned into a plasmid library and expressed from different promoters and ribosome binding sites as a fusion with proteins known to increase solubility. Since no cellular UP could be obtained, an NMR-based activity assay was established with two known thermolabile UPs. This assay allows one to follow UP activity even within cell extracts, which might be useful for the screening of large libraries generated by random mutagenesis. In order to obtain insights into the structure of our Pernix UP and two modified variants, Mr. Castelline has used simple modeling tools available via public servers. The results provided interesting information which will allow one to start with protein engineering even in the absence of an X-ray structure. For the most part, the language of this thesis is precise and the data are well presented. The description of the gene libraries which were created on the scaffold of ATIM is somewhat short and incomplete. In the public defense, Mr. Castelline has demonstrated his familiarity with all subjects of his thesis. He was able to provide convincing arguments to support the conclusions of the work and questions asked about mostly all aspects of his work. He replied clearly and logically. From my point of view, the candidate has made a successful defense of his dissertation, and I propose to the faculty that the thesis should be accepted.